Hello, dear friends. Today I'll tell you about the main processes and causes of aging known to date. The good news is that by knowing these causes, we can influence some of them right now. So what are the signs of old age? For some people, old age is associated with buying a bookcase for their iPhone. For others, with memory decline and stiffness of movement. Subconsciously, we can all determine a person's age with some accuracy. From a scientific point of view, however, it's not so simple. For example, stiffness of movement in a young person may be related to Betteredge's disease. A student may have a poor memory, while an elderly faculty member may remember all students by name. Poor memory cannot be a primary sign of old age. The sign of old age must meet three basic criteria. First, the sign of old age must appear during normal aging in everyone. That is, it must be typical. Second, its experimental enhancement must accelerate aging. Third, its experimental weakening should slow down aging and, consequently, increase healthy life expectancy. The first two criteria are easy to verify in an experiment. The third criterion is more difficult to verify. Human life is too long for us to accumulate enough data. Over the last 30 years, scientists have done a lot of work and have been able to identify 9 main signs of aging. The first sign of aging is genetic change. Our genome is more than just a long sequence of DNA letters. The strands of DNA are wound around coils of protein called histones. Various chemical buttons, handles, and levers can be attached to the DNA and histones to help turn genes on and off. These buttons, handles, and levers make up your epigenome. Your epigenome changes as you age. Levers are lost, inappropriately added, or moved. As a result, the precise coordination of gene activity can be disrupted. One particularly well-studied group of molecules that affect the epigenome are sirtuins, molecules that remove one type of epigenetic marker. Interestingly, our epigenome can be modified by diet, other lifestyle factors, or pharmaceuticals. Evidence that the epigenome affects aging comes mainly from studies conducted on yeast fungi, worms, and flies. However, dietary restriction in mice slows epigenetic changes, and when mice are deficient in one of the sirtuins, they show signs of accelerated aging. Moreover, when sirtuin is in excess, mice live longer. The second factor is the loss of proteostasis. The main job of genes is to make proteins, which are the heart and soul of cell biology. Proteins regulate virtually all chemical reactions and provide structure to cells. Protein homeostasis, or proteostasis, is the maintenance of all proteins in their original form and abundance. Proteins must be folded into precise and complex shapes, like origami, to perform their functions. However, as we age, proteins become damaged and begin to fold. The folded proteins not only fail to fulfill their function, they can become toxic. Alzheimer's disease is an example of an age-related disease caused by improper protein folding. The third factor is impaired sensitivity to nutrients. All living things, including humans, grow and reproduce when food is abundant. When food is in short supply, the body focuses on maintaining life support, thus increasing lifespan. In other words, live fast, die young works when food is abundant. Lack of food, on the contrary, prolongs life. Studies have been done on how the body responds to blocking the signals of nutrient excess. The first study is a calorie reduction diet. The second was tricking the body with the drug rapamycin. The body was made to think that there were not enough nutrients. And the third was blocking the action of insulin and its relative, insulin-like growth factor. All of these actions increased longevity and improved health in mice. The fourth factor is mitochondrial dysfunction. Mitochondria are inclusions in cells where energy is produced. A side effect is the production of free radicals and reactive oxygen species. For a long time, it was believed that free radicals were harmful to the cell. Now the opinion has changed. Free radicals are necessary for proper cell activity, but they must be in strictly defined amounts, neither more nor less than the golden mean. With age, mitochondria disrupt the production of free radicals, which is the cause of aging. The fifth factor is cell aging. Cells that used to divide actively and then stopped doing so but did not die are called senescent cells. As we age, the number of senescent cells accumulates. These cells release chemicals that affect the surrounding tissues and cause, in turn, the cells of the surrounding tissues to age, meaning they infect the adjacent tissues with senescence. Experiments on mice from which the old tissue has been removed have shown that these mice live longer and are healthy. The sixth factor is the depletion of the stem cell supply. Our ability to repair damaged tissue is critical to our health. This ability depends on the presence of healthy stem cells. Stem cells are a resource for repair. They are present in almost all organs and tissues. Healthy stem cells divide only when needed. The ability of stem cells to divide decreases with age. Stem cell function can be reanimated by external stimuli. Under experimental conditions, stem cell activation leads to better health and longer life expectancy. The seventh factor is the disruption of intercellular communication. Intercellular connections and interactions are very important in maintaining health. Hormones are an example. Hormones produced in the brain can affect the function of cells throughout the body and vice versa. The liver can send signals to the brain to reduce hormone levels. Nerve cells that perceive pain can signal the immune system and activate it throughout the body. As we age, these connections are disrupted. These disruptions lead to chronic inflammation that wreaks havoc on all organs and tissues. Although the cause of chronic inflammation in old age has not been discovered, research has pointed to the role of aging cells in causing it. The eighth factor is genetic instability. Each cell in our body, except red blood cells, contain DNA, providing the vital functions of our organism. However, genetic material is constantly attacked by external and internal environmental factors. As a result, damage accumulates in it. Just as when you rewrite a text, you gradually change one letter at a time. Eventually, the word may change beyond recognition. That's what happens to DNA. The cell normally has specific systems that repair the damage, just as proofreaders correct errors in a text. As we age, the errors become too many, and the systems for fixing genome mistakes can no longer cope, so genetic damage accumulates. That's how cancerous tumors arise. Measures to reduce DNA damage have been shown to prolong life. One example is a calorie-restricted diet. The ninth aging factor is telomere attrition. Telomeres are sections of DNA at the ends of chromosomes that protect the genetic material. Telomeres carry no hereditary information and are shortened at each cell division. The length of telomeres is such that the cell can divide a certain number of times and then age. Thus, the telomere length limits the number of cell divisions and determines the cell's lifespan. Telomeres are not shortened in stem cells because they have the enzyme telomerase, so the stem cells can divide indefinitely. In the experiment, animals with shortened telomeres live shorter, and those with lengthened telomeres live longer. So, we've learned about the 9 factors of aging. Now we know what we need to address to prolong life. Some of the factors can be adjusted very easily, but others can't yet be adjusted. Thank you for your attention. Always for you, Dr. Popmed.